Is it possible that we could, with gain of function research, create a virus that is more dangerous than previous viruses were?
We'll never know.
Because to me, it seems like a
We'll
real
never know.
possibility.
We'll never know the answer to that question because we'll never release it, and you'll never find out.
We'll never release it on purpose. So now I'm getting
on purpose. Now you're getting conspiratorial on me.
Well, it's no. I'm not conspiratorial. I just know how much human error is a factor.
No, of course you're allowing for that.
You can break a petri dish full of a dangerous virus.
Give you the example. The example that I will give you is smallpox.
Yeah.
So we've known about smallpox a long time. We've been able to raise smallpox in the lab. We've been able to store it. We've been able to ship it. It's very infectious. People die from it. And yet, there have only been 3 laboratory accidents with smallpox virus ever.
We know of.
2 of them occurred. No, we know this for sure because smallpox is gone now. There's no more smallpox in the world. Nobody catches smallpox because there isn't anymore. It's one of the few viruses that we actually got rid of. But people are still secretly working on smallpox virus. And you would know it in a minute. You would have people infected. They would spread the virus. People would become more infected. And eventually say, there was an outbreak. And you could say exactly where that occurred, okay? So that's what happens when you get an accident in the laboratory. So there was a laboratory accident recently in England about 10 years ago. That's recent, where an investigator made a mistake. The virus actually escaped from his laboratory in a stream of liquid. He threw it down the sink, which is stupid, and the sink was leaking, and the leak transferred down to the laboratory of another person on the floor below, and she caught smallpox and died.
Wow.
That's death number 1. Death number 2, he committed suicide as the result of his act. Those are 2 deaths from smallpox that were released from a laboratory accident. That's the kind of thing you're dealing with here. So the question that you've asked is valid, but we're now looking at the 1918 flu virus, which killed 2% of the world. And we're asking the question, why? The flu viruses that we have today don't do that. We have never had an epidemic as serious flu influenza as we did in 1918. It was called the Spanish flu, but it was a horrible name to give it because it was everywhere. So now we've dug up, how do you even get the virus to look at it to see what it was like? And it turned out to be very easy. In Norway, they buried a bunch of people who died from it in the permafrost, and their bodies were preserved. And the virus was still infectious that they could isolate from the bodies when they dug them up. And they then infected cells in vitro in the laboratory with that virus, and they sequenced the entire virus. And now they know exactly the region that changed to make it the most dangerous flu virus in the world. Now, isn't that worth knowing?
Yeah, but
they didn't do any gain of function studies. They just wanted to know why this virus was so dangerous.
Okay.
Now you're asking, can you make it even more dangerous?
I think they could.
Kill off 100% of anything.
I think they could. I think they. I mean, I know
you do because you don't know anything about DNA. That's why.
You're right. You're right.
Or evolution. Evolution doesn't work that way. That's not how evolution works.
It's always a percentage of people who are different.
Yeah, there always got to be a mutant out there that could resist everything. I'll give you the best example that you could probably know that you could relate to. The AIDS virus is a horrible virus.
Yeah.
It's slow. It's very difficult to detect before it's already caused its damage.
But there are people who are immune to the AIDS virus.
That's right. Totally. And they know exactly why, too.
But, but AIDS still kills 99% of people.
Oh, no question. No question.
So
what if you could make a vaccine based on the difference between the ones that live and the ones that would die from this? Which you could make a vaccine. Right? Well, we're trying. We're
trying it. But here's
we haven't succeeded yet.
I mean, I don't know my stance on gain of function research. I don't think I know enough about it. But isn't
Neither
it
does
possible?
anybody else. We haven't done enough. We haven't done enough of it.
Yeah. But I mean, devil's advocate, isn't it possible that somebody could potentially create a new AIDS virus? And, this is the Swiss cheese model. Somebody's got to fuck up multiple times here. But what if they make
I'm
an
turning
AIDS
off my
virus?
camera for a moment.
Oh, yeah, sure. No worries.
We're pausing, as it were.
Oh, you want to pause the video?
No. I'm going to just keep talking. That's
Okay,
it.
cool.
Go ahead.
So what if somebody creates something like an AIDS virus that has a 99% kill rate, and then also
Wait, whoa. How would they do that?
Through,
Tell me how you do that.
They take a pangolin DNA. Yeah, a virus, a pangolin.
But you have to know,
and then
you
they
have
say,
to know how the work works. You need an animal.
Okay, let's say they take the AIDS virus from a monkey, the rhesus monkey, and then they say
that's a simian virus.
Yeah, and then they say, how close can we make this AIDS virus from this rhesus?
They've been trying that experiment for 25 years without success. It's a difficult virus. It's got lots of parts. It's got huge numbers of genes. It's got a complicated life cycle. It doesn't revive around a spike. It's got two different parts to the virus that actually need to attach to the cell in order to get a cell to take it up, and it only infects B cells.
Do you think there's almost no chance that we could accidentally release, through gain of function research, that we could accidentally release a dangerous virus to the world?
You mean like Dr. Mengele is sitting in his laboratory in his basement and
because there's two different categories.
Look at all the technology that's necessary just to work on viruses.
Well, there's two different categories here, right? Dr. Mengele would be an intentionally malicious actor. So obviously,
Correct.
I think we should all be against intentionally malicious actors.
Yeah, but you're not going to stop them just because you're against them.
But yeah,
otherwise Putin would be bombing the hell out of Ukraine, right?
But gain of function research, I think, would be categorized as something different, right? Gain of function research is not intentionally making viruses to kill a certain population. This is, I would say, it's something like manipulating viruses in order to solve the problem. And the question is not, should we be intentionally doing genetic warfare? The question is, should we be researching viruses and changing them in order to figure them out better? Is that correct?
Yes, but you're asking the same question as someone would say, well, why don't we only write sonnet poetry? Why do we have to write iambic pentameter poetry, which is more difficult to pronounce and not a lot of people read it and that sort of thing?
Yeah.
Why would anybody try to prevent anybody from exploring natural process? Because that's what you're doing.
Well,
science is,
the answer is because
science is curiosity driven.
Iambic pentameter poetry doesn't spread smallpox to people. And I'm not against it, but I'm arguing devil's advocate here.
Neither does gain of function research. Because in the most cases, they're not even studying human viruses. They're taking a fish virus and they're studying that as to whether or not it can be more infectious for a zebrafish or not. But that's not going to hurt anybody. So if you limit it to non-human viruses, let's say that you were the oversight board for the National Institutes of Health, and you're a bunch of really studied scientists, you've all done bench research, you know what it's like, and you know it's necessary. And you could say, well, I think we'll restrict gain of function research to non-human issues.
Is that what they do?
No, but if you say that, then what happens? You learn all about nature except the part that you're really most interested in.
Yeah.
What will the next pandemic look like? Where will it come from? And how can we prevent it?
Yeah.
And if you don't do gain of function research, then you're never going to find out that answer.
So it's your argument that gain of function research is
you will live by the seat of your pants from nature supplying that answer for you, and the answer comes too late.
So I think a lot of the things that people are against in gain of function research is, I think they're conflating these scientists who are trying to prevent the next COVID with.
You are right.
With Mengele.
You're absolutely right, because Mengele could not have done that. In fact, what you're suggesting has never been done. We've never created a super pathogen.